Today we will be reviewing chicken egg white, Hatching of a New Old Biomaterial by Sherbrooke et al. In 2020, reviewed by Aditi Pattabhiraman and Jonathan Lerner.
Egg whites were long thought of as a cheap and abundant ingredient used in cooking for its binding and emulsifying properties. These same properties make eggs a useful and cost-effective biomaterial with biomedical applications. There are proteins present in the egg white that can be used as biomaterials, and these proteins are ovalbumin, which is the most common protein in the albumin, which is the egg white, and is responsible for the emulsifying process of them. Quinalbumin gives the albumin some structure and has some antimicrobial properties. Albomucin makes up a small 3.5% of the egg white, but gives the albumin their gel-like structure. The lysosome has the strongest antimicrobial properties and only makes up 3.4% of the albumin. The ovomucoid is thermally resistant and only makes up 2% of the egg. These proteins can be used to change their morphology, which means change their size and shape and structure. And some of these are, you could change it into a sponge, which is derived from gelatin and albumin and can help support tissue growth. Hydrogels, which are elastic and self-healing gels made from egg white that can be 3D printed into human tissues like ears. And microparticles, an easily manufactured material that can help deliver drugs orally. Some other morphologies are film, which are easily manufactured and are useful for implantable electronics because it mimics human tissue. Nanogels, which is egg white-based gel that's slow to metabolize, making it good for the delivery of hydrophobic drugs that need to be delivered over a long period of time. And a nanofiber, which when combined with gelatin can form a biodegradable nanomesh that's good for packaging food.
Egg white proteins are useful in vitro cell cultures, which are cell cultures that take place outside the typical biological environment. Egg white proteins have been shown to increase the viability in specific cell cultures and increase the proliferation of certain cells. Its low cost and availability makes it especially useful in laboratory settings. Amino acids, such as tryptophan, that are present in egg whites can help aid in the various healing processes. In addition, egg whites provide supporting material for tissue regeneration and other similar processes. Egg whites have been proven to be highly controllable, capable of loading sufficient amount of drugs, and made monitoring their subsequent release possible when part of a specific drug delivery system. These have also been proven to work in commercial settings. Biomaterials that are used for packaging should be able to account for factors like humidity and temperature. Egg whites, along with other additives, are good candidates for this. When polysaccharides are added, the water uptake increases, allowing it to adjust for humidity and temperature. Eggs are both very nutritious, but can be used with other foods as a powder for storage and transport purposes. As mentioned previously, combining egg whites with various polysaccharides can enhance its natural properties and create new properties within food, such as aerated confections. Biosensors can detect food contaminants and bioactive nutrients, and egg whites aid in the development of this technology. As we see, the egg, especially egg whites, is a useful, versatile biomaterial that is cost-effective and available, making it applicable in numerous biomedical fields.